Have you ever wondered if we could actually slow down aging? Well, today we're diving into some absolutely incredible new research that might have found a way to do just that, and it involves hunting down the secret villains responsible for aging, right inside our own bodies.
It sounds like something straight out of science fiction, doesn't it? But get this, our bodies are actually filled with something nicknamed zombie cells, and a lot of scientists believe they are a key reason we get old. So the big question, the one we're exploring today is, what if we could finally hunt them down and get rid of them for good?
All right, so before we get to the solution, we first need to meet the villain of our story. In the biology of aging, this troublemaker has a proper name, the senescent cell.
So these are senescent cells, but honestly, zombie cells is the perfect nickname. And here's why. These are damaged cells that are supposed to die off through a normal process, but they don't. They just stop. They stop dividing, but they refuse to die. So they just hang around in this weird undead state, causing all sorts of problems for the perfectly healthy cells around them.
And they cause a ton of trouble. The older we get, the more of these things we collect. And they're not just sitting there quietly. Oh no. They're actively pumping out this toxic mix of inflammatory signals. This creates a kind of chronic low-grade inflammation throughout the body, which scientists now link to a whole bunch of age-related diseases. So getting rid of them could be a total game changer. But there's a huge catch. They are notoriously difficult to find. And this quote from the research paper itself really hits the nail on the head. There's no single universal sign that just screams, "Hey, I'm a zombie cell." It's like trying to find a spy who is an absolute master of disguise. You have to look for a whole combination of subtle clues, which makes targeting them from removal almost impossible. Or at least, it was.
Okay, so this is where the science gets really, really clever. To find an enemy that's so good at hiding, these researchers launched what is basically a molecular manhunt, searching for one unique marker that would finally give these zombie cells away.
What they needed was a kind of molecular GPS system, a tool that could scan the surfaces of all our cells and lock on only to the zombies while totally ignoring the healthy cells, even if they're right next door.
So how in the world do you build something like that? Well, they use this brilliant technique called SELEX. You can see the idea here. Imagine you have a treasure chest filled with literally trillions of tiny, randomly shaped DNA keys. The goal is to find the one special key that fits a unique lock that's only found on the surface of a zombie cell. And SELEX is the super clever sifting process they use to find it.
So here's how the hunt goes down. First, they take that massive library of DNA keys and they expose them to healthy cells. Any key that sticks gets thrown out. We don't want those. Then, they take all the keys that are left and introduce them to the zombie cells. This time, any key that sticks is a winner. That's a potential tracker. They collect those winners, make millions of copies, and then they repeat the whole cycle over and over, getting more and more specific with each round.
And you can see right here just how well this worked. This chart shows the recovery, basically how many of those DNA trackers they found that successfully latched onto the zombie cells. After just 2 rounds, the recovery is pretty low, but look at that jump by round 8. They had successfully filtered out all the duds and zeroed in on a handful of DNA molecules that were fantastic at finding their target.
So, they'd found their trackers, the hunt was a success. But that just led to the next big question. What exactly had they found? What was this unique lock on the zombie cell that their new key was fitting into so perfectly?
And here it is, the money shot. They took one of their best DNA trackers and applied it to cells in a dish. In the columns on the left, you're looking at healthy cells, and you can see the tracker mostly just ignores them. But look at the columns on the right. The senescent zombie cells are lit up like a Christmas tree. This was the beautiful visual proof they were hoping for. I mean, that visual confirmation was incredible, but it still didn't solve the mystery of the lock. For this tracker to be a truly useful tool, they had to identify the exact molecule on the cell's surface that it was binding to. Using some really advanced techniques, they finally got their answer. You can think of it like this. They used their DNA key, which scientists call an aptamer, as bait to go fishing for the lock. And one protein consistently showed up again and again, fibronectin. The data here suggests that zombie cells display a specific version of this protein on their surface, making it the unique marker they've been hunting for all along.
You know, finding a target in a petri dish is a huge step. But the real test, the one that actually matters for our health, is proving it works inside a living, breathing organism. It was time to see if this tracker could find its target in mice.
The researchers looked at lung tissue from mice at different ages, and the results were just remarkable.
On the right, you can see tissue from a young mouse, just 3 months old, with almost no green signal from the tracker. But look down the column as the mice get older. At 22 and then 30 months, the tissue is just glowing. This perfectly mirrors what we already know about aging. The older you get, the more zombie cells you have.
But to be absolutely 100% certain, they designed the ultimate test. They used a special, genetically engineered mouse where they could basically flip a switch and eliminate most of its zombie cells. This set up a really simple question. If the tracker is really finding zombie cells, its signal should pretty much disappear when those cells are gone, right?
And the answer was a resounding yes. In the images on the left from an untreated mouse, the tissue is glowing brightly with that green tracker signal. But on the right, after the zombie cells were cleared out, the signal is almost completely gone. This stark difference is the smoking gun. It's the proof that the tracker is finding exactly what it's supposed to find and nothing else.
And the hard numbers from the experiment back this up perfectly. This chart just quantifies what we just saw in the pictures. The fluorescent signal from the tracker is high in the untreated mice, but it plummets after the treatment clears out the senescent cells. It's really that simple. No zombie cells, no signal.
So this is all fascinating science, but what does it actually mean for the future of medicine? This discovery isn't just some academic exercise. It could open up a completely new frontier in how we think about and treat aging itself. The potential applications here are just enormous. I mean, imagine being able to measure a person's zombie cell burden to see their true biological age. This could help predict who's most at risk for certain diseases. But what's even more exciting is the idea of treatment. You could attach a cell-killing drug to these trackers, basically turning them into guided missiles that seek and destroy only the zombie cells, while leaving all the healthy tissue unharmed. So, it all leads to this final, tantalizing question. We started with this problem of hidden zombie cells, we followed a super clever molecular hunt to find them, and we ended with a tool that actually works in a living animal. This research gives us a powerful new weapon in the fight against aging, and it really makes you wonder, could these trackers be the key to unlocking not just a longer life, but a much, much healthier one for all of us?